All right, so ladies and gentlemen, we are going to be taking a look at cell division. So we are continuing our conversation about genetics, but specifically we are going to be looking at genetics from the cellular level. Before we can have a conversation about genetics at the cellular level, we need to first talk about cell division. There are two forms of cell division. There is mitosis with a T, that is how we make diploid cells. And then there is meiosis with an E, which is how we make sex cells or the haploid cells. So, okay, as we go through this, I will kind of tell you which of where we're at in the actual assignment in terms of the handout. Okay, so let me just get there really quickly. Let me just stop this. And I just need to open up my assignment. Give me 2 moments to do that. Nope. Okay, that's not going to work right now. That's fine. No worries. All right, cool. So we're just going to go ahead and get this done here then. All right, so ladies and gentlemen, let's go ahead and move forward. So at the top of your sheet, it asks 2 questions. Number 1, why do cells divide? And number 2, instead of dividing, why don't cells just get bigger and bigger and bigger? I don't remember if it was in the video that we watched yesterday, but I do know that there is an Amoeba Sister video where it talks about cell division, and I think we're watching that tomorrow. But why do we need to divide cells? Why don't cells just get bigger and bigger and bigger? Well, it's pretty simple. There's 2 main reasons as to why there is a limit to our cell growth. Number 1, cells divide rather than continuing to grow larger and larger. So the question becomes, why is this? Okay, we want to know why this is. It's pretty simple. The larger a cell becomes, the more demands it places on the DNA. Please do keep in mind, ladies and gentlemen, that DNA is pretty much the instructions for everything in the cell. So again, it's kind of like, and I always tell people, sort of think of this as if it was like a teacher in class. If a teacher has a small classroom, let's say, for example, 10 students, if every student has a question, it's really easy for that teacher to get to every single question that every single student has because there's only like 10 of them. However, imagine that same teacher, again, like we only have one set, well, actually we have 46, but you only have one full set of DNA per cell, right? Imagine that same thing as if a teacher had 100 students in a class, and every single one of her 100 students had a question. They had a demand of her. In a regular class period, there's no way that that teacher is going to be able to get to every single question, because even if she spends only 60 seconds, 1 minute on each individual student, we're going to run out of time. So as a cell becomes bigger, there are more demands placed on that cellular DNA, and it becomes really hard to manage. Also, if cells get too large, those cells are going to have a really, really hard time moving nutrients into the cell to help nourish that cell. It is also going to have a hard time getting waste products out of that cell. Because if you recall, when we talked about cells, I want to say during first card marking, maybe it was the very beginning of second card marking, I don't genuinely remember. One of the conversations we had was about the cell membrane, and we said that there is active transport and passive transport, and no matter what the type of transport it is, in order to get things in and out of the cell, it has to go through the cell membrane. And there's only a few ways to do that, and a lot of things are limited in terms of if it's active transport or passive transport, how these things get across our cell membrane. So these are the 2 problems that arise when cells get bigger and bigger and bigger. Too many demands on the DNA, and too difficult to get nutrients in and waste products out. All right, problem number one, our DNA does have limits. As amazing as our DNA is all the information that our cells needs to function is always stored in your DNA. Everything that makes you you, every function that your body does, whether it's breathing, whether it's digesting your lunch that you just had, whether it is breathing, it doesn't matter what it is. Every single thing that you do, everything that any organism does, that information on how to do it, the how-to manual is in your DNA. Now, your DNA inside of your nuclei is packaged into chromosomes. I always like to think of the chromosomes um, as sort of having like really, really curly hair. Some of you guys have really curly hair. And if I were just to look at you, maybe when your hair is in its natural state, or maybe like just after you finished washing it, your hair might seem kind of short. But then if you grab your hair and you pull it out straight and you let it, the curl come out, your hair is actually very long. I know you guys know what I'm talking about. That's like DNA when it gets packaged into chromosomes. DNA is a long linear molecule. But when we package it into chromosomes, what we do is we twist it up tighter and tighter and tighter and tighter. And we make these tight little packages of chromosomes. So it's kind of like your hair. If your hair were straight, it's very, very, very long. But when we allow the curl into your hair, that natural curl that many of you have in your hair, the hair shortens up. And it's not that the hair is actually short. It's just that the length gets a little bit shorter because it's so tightly curled up. That's exactly what happens when DNA is packaged into a chromosome. It just is tightly, tightly wound up. Every chromosome is made up of DNA, and that DNA has hundreds and thousands of genes. Each of your genes is a set of instructions, and it makes a particular protein. Now, some of those instructions we already talked about when we talked about the Mendel genetics. Some of those instructions are the instructions to make your eye color. They're the instructions that make how wide your nose is, how round or how almond shaped your eyes are. They're the instructions that tell your body, okay, make attached or detached earlobes, right? These are the instructions that say that you are going to be tall or you are going to be short. But some of these instructions really are just to make proteins that help in other areas like digestion or breathing or, any of your many, many millions of functions in your body. Your cell is always making copies of your DNA. It's always making copies of these genes. And the reason why you're always constantly making copies of the genes is because what ends up happening is you always have to make these proteins, right? You have to make your body's always breathing or digesting or doing something. So you're always going to be making copies of this instruction manual to go out to the ribosomes. If you guys remember when we talked about cells, we said that ribosomes are where we actually build proteins. It's like the protein factory of the cell. When cells are small, there's enough information stored in the DNA and it can adequately see to the needs of that cell. Much like, again, when you have a small classroom and everybody has a question, the teacher has no problem answering all those questions. She can see to the needs of her students. When a cell is small, the DNA can see to the needs of the cell. However, if a cell gets too big, then the DNA cannot meet the needs of the cell because there are more and more demands when there's a larger cell. Just like when a teacher has a really, really large classroom and everybody has a question. If I had 200 kids in class, there's no way if all 200 of you had a question, there's literally no way in an hour I would be able to answer all 200 questions. There are simply too many demands being placed on that one teacher. Just like if that cell gets too big, there are simply too many demands being placed on that one set of DNA. All right. Second problem, a growing cell needs more food. And it's not like food is just going to magically appear out of nowhere. So here's the deal, guys. and I'm just going to kind of go through this, and you guys can fill in your notes as you need to. A cell is always taking in nutrients, and those nutrients might be food, it might be water, it might be oxygen, and that all of those nutrients have to move across your cell membrane. Now remember, we have active transport, we have passive transport. There's different ways of getting this stuff into and out of the cell. But either way, we're always taking in nutrition. At the same time, we are always getting rid of waste products. Whatever waste products your cell creates have to leave the cell, because if they don't leave the cell, that can be toxic for the cell. It can actually kill the cell. Interesting side note, right now, if you breathe out all that carbon dioxide that you just breathed out is waste product from your cells. Because when you go through certain processes, the waste product is carbon dioxide gas. That's what you breathe out. Eventually, it has to make it out of your cells. So every time you breathe out, you're actually breathing out cellular waste product. So they leave the cell, and then it leaves your body. A larger cell requires way much more food, way much more oxygen, way much more water. A larger cell also generates a lot more waste. Okay, these are problematic. Why? Why is this problematic? Well, because, ladies and gentlemen, as a cell grows, if a cell gets too big, then the volume of the cell changes much more faster than its surface area. And the surface area basically is your cell membrane. Keep in mind, ladies and gentlemen, everything in your cell that comes in, everything that goes out has to pass through the cell membrane. That's the whole reason the cell membrane is there, is to help maintain homeostasis. That becomes a problem, because if the cell gets entirely too large, then the membrane, the cell membrane, that surface area is not adequate. It is not enough to transport in the large quantities of food, the large quantities of water, the large quantity of waste products that we need to get into the cell and out of the cell. We just don't have enough space. It's kind of like if Amazon delivery only wanted to use 2 trucks. It just wouldn't work at all, because there's way too much stuff going in and out of Amazon to only have like 2 or 3 trucks. They have like millions of trucks. Much like your cell, if I only have one really large surface area, but inside the volume is entirely too big, that's not going to work. All right. So your next section involves these areas that I just popped up. The size of a cell is limited by its surface to area volume ratio. All right. That's what limits how big your cell can be, is the surface area to volume ratio. All right. Basically, each cell needs to have the highest surface to area volume ratio that is possible in order for this cell to function at its peak performance. Now, I know you guys do not have this chart in your notes, and that is perfectly fine, but I just want to do a quick little bit of math, because I love math, and well, I'm kind of a nerd about math. All right, so let's look at the small cell on the top of the chart. And to make life easy, I have literally just made these cells into cubes, because that just makes the math way much easier than trying to do spherical math, because geometry, yo. All right, so surface area is length times width times the number of sides. So in this case, it would be length times width times 6 sides in a cube. So length times width, 1 times 1 times 6. This has a surface area of 6 square centimeters. All right. Volume. Well, volume is length times width times height. So the volume for this cube would be 1 times 1 times 1, which is a volume of 1 cubic centimeter. To calculate my surface area to volume ratio, I literally just compare the two and then reduce the fraction. So this is a 6:1 ratio, or I could say a 6, like 6.1. Okay, cool. So that's, 6:1. That's pretty decent. Let's look at that next cell. All I did was double the size. I added one more. So it has a length of 2, a width of 2, a height of 2. Same math. Surface area, length times width times 6 sides. So that would be 2 times 2, which of course is 4. 4 times 6 is 24. A surface area of 24 square centimeters. Volume, same thing. Length times width times height. In this case, 2 times 2 times 2, which is a volume of 8 centimeters cubed. All right, let's take a look at the ratio. This gives me a ratio of 24 to 8. If I reduce this, it's a 3:1 ratio. My ratio has gone down, right? A little by, about half. Ugh, that's not good. Because again, my cell is going to function better the higher this ratio is. So, I mean, I don't know if this is necessarily true or not, but you could sort of make the assumption that if it's half the surface ratio, you can say with some certainty that the cell is only going to function half as well. Okay. Obviously, that's not 100% true, but it's an assumption that we can make. That being said, let's take a look at the third cell. All right. So for my third cell, again, all I did was add one more to my dimensions. So now I have a length of 3 times 3 times, surface area of length times width, which would be 3 times 3, which is 9, times 6, of course, is 54. So 54 square centimeters. For the volume, I'm going to go length times width times height, 3 times 3 times 3, which of course is 27. So now my ratio is 54 over 27, or a 2:1 ratio. The bigger my cell gets, the smaller the ratio becomes. This is just math, guys. And we know that the bigger this ratio is, the better the cell functions. Because again, those two limiting factors. Number one, the DNA doesn't see to the DNA cannot see to all of the functions. And secondly, the cell membrane has to be able to get the nutrients in and out, and that just becomes more difficult the bigger it gets. All right, so we are on the next page now, which is the second one where it says cell division. All right, so this is part A. Cell division is the process by which cellular material is divided between two new daughter cells. All right, so this is letter B. The mother cell divides down to form two daughter cells. Which of course those two daughter cells will be identical to each other and the mother cell. When I have this kind of cell division, ladies and gentlemen, this is sort of like what we call when we talk about cloning. You have two identical daughter cells that were formed from the original parent or mother cell. We are moving into, I want to say this is part C or letter C. Before the division begins, the cell first must copy all of its DNA. It has to make a copy of every single last one of its chromosomes. Because again, all of your information that is going to help to keep you alive, all of your information that to build any organism or to keep any organism functioning, of course, is needed from that DNA. I want to say this is letter. Each daughter cell is going to get a complete set of chromosomes. All right. Let me get this down here. Sweet.
Miss Cano-Michael,
Yes,
can you
love.
go back to the last slide so I could take a picture? Because I missed that one.
I am making a video copy of this, so you'll have access to that YouTube video as soon as I'm done with this.
Okay.
All right, cool. All right, each daughter cell is about half of the size of the parent cell, which is important because, again, the idea is we are talking about making the cell smaller. Cell division basically makes smaller cells. Now, of course, the problem is as soon as you divide the cell, it starts to grow again. And so your cells are constantly going to be dividing. Okay? So each daughter cell has, this is letter D, an increased surface area to volume ratio. And remember, the idea is that we want to have the largest possible surface area to volume ratio, because the larger that surface area to volume ratio is, the better our cell is going to function. Okay? All right. This is part 3, where it says chromosomes during eukaryotic cell division. And we are just going to get through this section. I think, oops, sorry, didn't mean to do that. So this is part A. Chromosomes are composed of DNA and proteins. The DNA contains coded genetic information of the cell. Again, your chromosomes are kind of like your hair, right? If you have naturally curly hair, your DNA and your proteins just wind up really super tight, and they make these packaged chromosomes. But again, those chromosomes are a lot of information. There's a lot of gene information there, a lot of instructions there. It's just in this nice small package. Again, your DNA is and again, this is one of those characteristics of all living things. All living things have DNA, which is the genetic information, which we can inherit from one generation to the next, which has all of the instructions for life in it. This is going to be letter B. Each cell must make copies of the chromosome before cell division occurs. Because remember, the whole point is at the end of cell division, not only do we want to have daughter cells, but those daughter cells have to be able to function. And without a full set of chromosomes, it's not going to be able to function properly. Each daughter, this is letter C, each daughter cell will get a complete set of chromosomes. Okay. Now, again, this depends on what kind of chromosomes are they getting. Are they getting a full set? Is it a haploid cell, or are they getting a half set of chromosomes? I'm sorry, I said that backwards. If it gets a full set of paired homologous chromosomes, we are talking about a diploid cell. If it's getting a full set but non-paired chromosomes, then it is going to be the haploid cell. All right, we are moving into letter D. The cells of different organisms have a different characteristic number of chromosomes per cell. For example, human beings have 23 pairs of chromosomes if it is a diploid cell, or if in a diploid cell, we would say you have 46 chromosomes total, right? So 46 chromosomes total, but they pair up. Now here we have a male and a female human being, and we can see those chromosomes in this karyotype, but we'll talk about more what that means in a minute. So let's take a look. What about other organisms? Well, what about fruit flies? What about crab? Fruit flies only have a total of 8 chromosomes, or 4 pairs. The red king crab has 208 total chromosomes, or 104 pairs. Okay. Each organism has its own number that is unique for that particular individual organism. All right. So some have more, some have less. Some that we think would be really simple have a lot. Some that we think would be really complex only have a few. Great example. I think dogs have almost twice the same number of chromosomes as humans do. Just FYI. So the number of chromosomes doesn't necessarily speak to the complexity of the organism. It's just what that organism evolved to. All right, moving on to letter E. Cell division occurs in two main stages. Number one, we have mitosis, which is the division of the nucleus. And then number two, we have cytokinesis, which is the division of the cytoplasm. Moving into letter F, chromosomes are not visible unless you are doing cell division. So look at this picture that I've included here, guys. In some of these cells that we are looking at, we can clearly see these sort of like rod-shaped objects. Those are your chromosomes, okay? And then in other pictures, like maybe this one over here, it just kind of looks like a single cell and this like kind of dark nucleus in the center. This one down here, I can see sort of two areas that have chromosomes in them. Over here, I see chromosomes. So again, your chromosomes are only visible during the process of cell division. If you are not going through cell division, then the chromosomes usually are not visible even under a microscope because like DNA strands are just too small. At the beginning of cell division, why this is possible is that the chromosomes condense even further. So if you recall, we said that chromosomes were already your DNA, nice and compact. Well, prior to cell division starting, they are going to compact even further, which is kind of cool if you think about it. All right, so again, big takeaway, cell division occurs in two stages, mitosis followed by cytokinesis. And again, the only time you can actually see your chromosomes is during cell division, and that is seen in this microscope picture. All right. This is going to be our last slide for today because we are getting close to the end, and I want people to have time to ask questions or simply to get caught up. So this is going to be letters G and H that we are looking at here. Before cell division takes place, each chromosome is copied or replicated. You will hear me say both words. At the beginning of cell division, each chromosome consists of two sister chromatids. I'll explain that in a minute. And these chromatids are connected in an area called the centromere. All right, so what the heck is all that? That's a lot of vocab all at once. So let's take a look at what all that means. All right, so here we go. This whole thing right here is your duplicated chromosome. So normally your chromosome, and let me change colors real quick, would just be this one thing. That is a single chromosome. All right, so that's your single chromosome. So before I'm going to divide, I have to double my chromosome so you can see where it's been doubled. So here is the other chromosome that just got copied, all right? So now we have a duplicated chromosome. When you have a chromosome that has been duplicated, we now call this whole thing, both of these sides are my sister chromatids, all right? So this is one chromatid on one side, the other chromatid's on the other side. They are now hooked together. They are stuck together by this sort of button-looking object in the center, and this is a centromere, which I just spelled wrong, but that's okay. All right, so ladies and gentlemen, here's where it gets weird. This picture that we're looking at is one duplicated chromosome, or it is a set of sister chromatids. But if I were counting this as a chromosome in my So I would only count it as one chromosome. And the reason why is because when I'm counting chromosomes, I'm actually counting centromeres. It's weird. I'm not the one that made the rules. It is just what it is. All right. So we are going to stop here for the day. It'll give people a chance to ask questions, as well as I can check up on my attendance, because I know I had let some people in later, or after the fact, I should say. All right. So